Good afternoon, good morning, good evening. Whatever time you are watching this video right now, I hope you've had a great day, great evening, great afternoon. This is the refresher for Unit 1.3, so bacteria and viruses. So without further ado, I'm going to get into this. You should be working on the Unit 1.3 virus notes. If you are not working on those notes, if you did not get everything in class as I was doing it, make sure that you have it and next to you right now that you're working on it. Okay. Any questions about anything, please email me or chat with me on Go Guardian if I am actually in class right now while you're watching this, or just send me a message in Canvas. Okay. Without further ado, let's begin. Okay, so Unit 1.3 viruses and bacteria, the evil within. Okay. What you see right here is an example of a bacteriophage. Okay. A bacteriophage is a organism that has, first off, you see right here is the virus. Okay. What this is a virus that has the genetic information, the RNA, the DNA inside of this capsule. And so what is occurring is that the virus has landed on the cell, and then throughout the tail, the virus has started to enter its genetic information into this cell. Okay. Once it does that, the capsule just blows away, and then a new virus comes, so on and so forth. Now, you may see something that I call a bacteriophage that looks exactly like this, okay? That is what vaccines appear when they are trying to mask as a virus and enter your system in order to keep you from getting sick. So, microbiology and evolution. So, major concepts focused on in this unit, 1.3. So, we're going to realize what viruses and bacteria actually are. We're understanding that they're everywhere and unavoidable. Okay, so my people who are clean and neat freaks, there's no way that you can escape this. Okay, ways that they can help us out in the world: gene cloning, genetic engineering, um, bioremediation, um, decomposition. So ways that without bacteria we would have no land plants. They're not, because bacteria are the ancestors of plants. Okay. They were capable of photosynthesis. So they put the first oxygen molecules in the atmosphere. We've learned that that was the great oxidation event. And they're the oldest of all the kingdoms. So there has actually been fossilized forms of bacteria that have been found, um, that were older than 2.5 billion years. You got to remember that the Earth is around 4.5 billion years old. One of the oldest bacterium ever found, I believe, and you can search it on Google if you like, around 3.8 billion years. So bacteria has been here for a very long time. So it is important to know that bacteria outnumber us, our human cells, 13 to 1, which means they are very effective and very efficient, viruses and bacteria, at getting in our cells and making us sick. So other prokaryotes called archaea are similar to bacteria and share many characteristics with them. These include lack of membrane-bound organelles, can live in warm places, and can survive without little oxygen. So these are the three shapes of bacteria. So you have cocci, bacilli, and spirella. Okay. So bacteria are very small. So when you look at a bacterium cell, compared to a plant and animal cell, they're very small. Okay, only 1 to 5 micrometers wide. Okay, when you look at a eukaryotic cell, which plant and animal cells are, those cells are 10 to 100 micrometers wide. Okay. And then we have ways that bacteria obtain food and energy. They live in different environments. So obviously, just like humans, we all live in different environments. So if we live in a colder environment, then we would what? We would eat the animals that lived in that colder environment. We would learn how to survive in those colder environments, just like bacteria do. Okay, you have some bacteria that feed on dead organisms. You have some bacteria that take nutrients from a living host, such as us, a symbiotic relationship, and then photosynthesis. So you got to remember that plant cells are the great, all those great grandkids of bacteria cells. So some bacteria can still photosynthesize. And so we've seen this in a few of the videos that we watched. Um, One group of bacteria are aerobic, okay, and then there's another group that's anaerobic. Aerobic means that they need oxygen to breathe. Anaerobic means that they do not need oxygen to survive. So, without further ado, what you see right here is a bacteria cell and a few of the organelles. So you have the bacterium, flagellum, the nucleoid. Remember, they don't have a nucleus. A nucleoid, which just has circular DNA in it. A capsule, which protects them. So the capsule, the cell wall, and the membrane all protects that genetic information in the inside. A cytoplasm to keep the ribosomes in order. So ribosomes right here, which are a form of protein, plasmid, and then pili, which helps them move around as well as the flagellum does. So, bacteria are microscopic prokaryotes. So, we learned that a prokaryote was a single cellular organism. Do not have a nucleus or organelles such as mitochondria, etc. We already learned that bacteria live almost on every surface of the earth, from its highest peak to the lowest sea level of the ocean. Okay. In order to survive these extreme environments, they have learned to adapt. All right. Some bacteria can live in the magnum of a volcano, which is thousands of degrees hot. And some bacteria can live close to a perfect vacuum, okay, of the deep ocean, all right, where no oxygen exists. And this is done with the help of endospores. So bacteria have a thick external wall around the chromosome and the cytoplasm. That endospore protects it from anything that is trying to kill it. That's why bacteria cells are hard to kill, because you have to eliminate that endospore in order to kill it. So let's keep going. So we know that Anton von Leeuwenhoek discovered prokaryotic cells by very unsanitary means in the 1660s. Remember, he looked at the gunk in his teeth, he looked at specimens of his feces, and he saw what he called animalcules floating around in those things. Okay. So in the 1850s, Louis Pasteur came up with a way to sanitize food and drink. So he came. So if you have ever been to the grocery store and you see pasteurized milk, pasteurized orange juice, pasteurized cheese, he's responsible for coming up with the procedure to clean food and water and get all harmful bacteria out. Okay. They're estimated 3.5 million years old. Okay. They're estimated to be that old. The problem with that estimation is that we don't have any fossils of anything older than 3.8 million years old, because everything disintegrates and everything ages and withers away with time. Okay. So, bacteria cell wall is surrounded by polysaccharides called the glycolyls. The one on the left is a well organized called a capsule. If it is loosely organized, it is called a slime layer. Okay. We already know that they're 1 through 5 micrometers, and we know that they move using their flagellum to spin 360 degrees to attach to a host cell. So it spins in a perfect circle to attach to the host cell. So, beneficial bacteria in nature. So we have some types of bacteria that are beneficial. Okay. We have some that help us with digestion. Okay. And prevents us from getting food poisoning, which is a very terrible thing to have food poisoning. The name of that bacterium cell is called lactobacillus. Okay. Also helps with the decomposition of breaking down dead organisms and organic waste. Nitrogen fixation is the conversion of atmospheric nitrogen compounds that are unusable by living things. That's also a part of something that you may have heard of in sixth grade, the nitrogen cycle. And bioremediation, the use of bacteria in order to clean up the environment. So when they have chemical or oil spills, they use a form of bacteria to help clean that up. We will watch this video at a later date. So, pasteurization. So, pasteurization is the process of heating food to a temperature that kills most harmful bacteria. Okay. So, all he did was he had two experiments. His hypotheses were bacteria arise spontaneously in the broth. Hypothesis B, bacteria in the air contaminate a broth. Okay. So his first experiment was he boiled his sterilized broth and then he briefly opened the flask. Okay. So he had an 89% show of growth. Then he boiled to sterilize broth and then flask was opened briefly again inside of a building. Okay. 32 more percent show of growth. So his conclusion, well, let's talk about the second experiment. So his second experiment, he sterilized the broth. He left the flask to open air. Okay. What he did was the next one is that as this broth was sterilized, there is pure air that is right there at the beginning of where it goes into the flask, but the regular outside air has entered here and the bacteria has collected here. So that means the rest of this is pure air, which does not affect the broth. Okay. So now let's talk about the two conclusions. Hypothesis B is supported because relative concentration of bacteria in the air explains these results. Let's go to the next one. Hypothesis B is supported because when air reaching the broth contains no bacteria, the flask remains free of growth. So, why is pasteurization important? Pasteurization is important due to a bacterium called botulism. Okay. Botulism is very extreme food poisoning. Okay. Um, it is what happens when you leave food out for a very long time. Okay. Without refrigeration and without other things to slow the rate of deadly bacteria growth on your food. Okay. Um, there is a way that you can get botulism from eating food that were in cans that were dented, because when the can was dented, that allows some oxygen to seep into the can, therefore contaminating the food. Okay. So what is botulism? So it's food poisoning. So it can cause muscle paralysis, shut down your respiratory system, and eventually death. There has been people that have died from food poisoning from very extreme cases of botulism. So cholera is another example of a bacteria. Okay, cholera is an infectious disease of the small intestine. Okay, The way that someone gets cholera is like this. Say that we all lived in a town, right? And we only had one river as a water supply, okay? And people use the bathroom, they relieve themselves in this water supply, all right? Well, you think, okay, fine, it's the water supply, nothing to happen because, water's running. Even though that is true, if the water is still running, the water is still cold, there's no way to sterilize the water. There is a way for that water to be contaminated with cholera if that person who relieved themselves in the river is infected with cholera or has the disease cholera. Okay. So when you get cholera, you have pain in your abdomen, gastrointestinal issues, nausea, severe diarrhea, vomiting or watery diarrhea, whole body dehydration, lethargy, which means that you're tired, or water electrolyte balance. Okay, that means that you're not getting the amount of electrolytes that you need to get from different types of liquids, and the salt is running out of your body. Okay. That's why some people, when you're sick, you drink Gatorade because that helps your water electrolyte balance. And this is just a political cartoon to show the effects of cholera. So in 1991, Peru lost millions in tourism after a cholera outbreak because if you can't drink the water, why would you want to go there? Okay. The Rwandan crisis. If you do not know what the Rwanda crisis is, I believe that you should look that up. That is a very important piece of not only African history, but world history. It was a very tragic event. Um, you should look it up, and you will see that almost 30,000 people died after the outbreak of cholera. They died because there's no way, there's no clean water supply. They're just running for their lives, and so when you're running for your life and you're trying to survive, you don't care about trivial things such as, is this water safe to drink? You're gonna see water, I'm thirsty, I need to drink. Spanish flu. And this one is important because right about now, COVID, even though COVID has turned society upside down and has caused a lot of heartbreak and has caused a lot of hardship with people. We're not in school because of it. COVID has still not touched a fraction of what the Spanish flu was. So, influenza. We should know influenza due to that what can kill bacteria lab. We should know about influenza, okay? Influenza is a virus. It's the flu. All right. Now, the flu happens every year. There's always a flu season. That's why it's always important to get your flu shot so you can be prepared for the upcoming flu season. Well, the Spanish flu was different. Okay. The Spanish flu, which was caused by H1N1, okay, with genes of avian origin. That means it came from birds. So the Spanish flu crossed over from birds to humans. Okay. There are viruses out there that can cross over from animals to humans. Okay. This is an example of one. HIV and AIDS is an example of one. There are mad cow disease. There are a lot of examples of diseases or things that can afflict us crossing over from animals to humans. Okay. So 1918, which I believe is, this is 2020, almost 102 years ago, or I believe so, 102 years ago. So the most severe pandemic you can think of. Okay. So from 1918 to 1919, all right, 50 million people died. Imagine being alive during that time. You already have I believe the Great Depression is going on from this time. World War I just ended, and now most of the world is getting over a war and getting over not having any money. Now you have a flu that comes around and it kills 50 million people with over 500 million individuals getting it. It is the most deadly pandemic that you can think of. COVID has still not reached these numbers. And the reason why it was so deadly because there were no vaccines. No one had a vaccine to treat a worldwide epidemic. Okay. So the way that we're trying to fight COVID is the way that they fought the Spanish flu. They quarantined, practiced good hygiene. You know, you're washing your hands. They wore masks. Okay. They didn't really shut down like all these different things like sporting events and different type of gatherings. They just made masks mandatory. So now that was the Spanish flu. So now we're going to talk about hantavirus. Okay. So mice are everywhere. Okay. Mice are everywhere, and mice are not clean animals. All right. You get hantavirus from inhaling or coming around urine or feces of a mouse. Okay. It can cause a hemorrhagic fever, which means you're bleeding from the openings in your body. It can be inhaled by cleaning up areas that mice have infested. That's why when you see people cleaning up areas that mice have infested, you see them with, the respirator mask on, okay? And there's no treatment. You just got to get rest. We'll watch this video later. So now, bacteria are prokaryotic organisms, meaning they do not have sex as eukaryotic organisms may have, which means that they reproduce asexually. Okay? Because they reproduce asexually, they reproduce in the following way: binary fission. Okay? Binary fission, the singular circular chromosomes replicates, and then two copies separate the cell into two cells. This takes around 12 minutes or so to complete. Mutations that can occur are passed on quickly and pushed out a genetic code, making a bacteria genetically adaptable to any condition. This is why bacteria can survive for a long amount of time, because if something can replicate so fast, then there's no way to catch up. Okay? There's no way to catch up with it. There's no way to quickly identify it once it gets in the body. That is why certain things are hard to combat, such as AIDS, because it replicates so fast. And through that replication, you hardly even know you're sick until it's too late. Conjugation. Okay. Conjugation is a form of DNA transfer between a donor and recipient cell. And through a conjugation tube, the DNA and the plasmid are both switched and shared between both cells until the donor cell and the recipient cell are identical and they have the same amount of genetic information. So, sexual reproduction in bacteria continued. So you have transduction, transformation, conjugation. We've went over conjugation. Transformation is just when a cell picks up something. Okay, so a cell can just be going around and just picks up a form of virus or a bacteria. Okay, and then transduction is why I showed you at the beginning. It is when that bacteriophage lands on a cell and it inserts all of the information needed. Okay, and then we have ways that bacteria metabolize. All right, photoautotrophs. That means they use photosynthesis. Okay. Um, They use solar energy to reduce carbon dioxide to organic compounds. They have two types: those that evolve first and do not give off oxygen, and those that evolve later and do give off oxygen. Okay. Chemical autotrophs. They use chemicals, just as their name said. These bacteria carry out chemical synthesis. They oxidize inorganic compounds such as hydrogen gas, hydrogen sulfide, and ammonia to obtain energy. So their main goal is to oxidize nitrogen. And now you have obligate anaerobes and facultative anaerobes, organisms that need oxygen and those that do not. Okay. Obligate anaerobes are unable to grow in the presence of free oxygen. A few serious illnesses, such as botulism, gangrene, and tetanus, are caused by aerobic bacteria. Okay. In contrast, facultative anaerobes are able to grow in the presence or the absence of gaseous oxygen. So now we have viruses. So West Nile was a big one, okay, carried by mosquitoes, and I forgot what year it was, but there was a pretty serious outbreak in this area of West Nile, and that you had to start wearing, long sleeve clothes, jeans, just to make sure, repellent, just to make sure that mosquitoes would not bite you. So viruses are associated with a number of plants, animals, and human diseases. So AIDS, rabies, polio, measles, chickenpox, mumps, flu, those are all forms of the viruses. Okay. Viruses also have RNA and DNA that's protected in a small capsid, and they only reproduce using the host material from the host cell. So, 1884, Louis Pasteur suggested that something smaller than bacteria was responsible for the rabies disease. And if you don't know rabies disease, it's a horrible disease that an animal can get. We usually associate it with dogs, and eventually it just takes over the nervous system and makes them very angry and violent. And now we have obligate intracellular parasites. All right. All this means is that viruses cannot reproduce outside a living cell. Remember, we talked about the 7 characteristics of life at the beginning of the year. This is the main reason why viruses are not considered living, because they need a host specific, which means they can only infect what they are genetically coded to infect. Okay. They need to specifically infect a specific thing. So if a plant virus was for a plant, such as a tree, a bush, etc., it can only affect that. Okay. Plants can't catch AIDS. Okay. There's no way for that to happen because AIDS is a virus that has been made for animals, such as us. Cats can catch AIDS. Monkeys can have AIDS. A few other animals can have AIDS. And then we had the lytic cycle and the lysogenic cycle. Let's see. Okay. The lytic cycle is just what I was showing you, how the bacteriophage drops on the cell, puts all this genetic information in there, and then it makes more bacteriophages, and then the cell bursts, releasing all the other phages into your system to infect all the other cells. And then the lysogenic cycle, this occurs when the viral DNA is integrated into bacterial DNA and then is passed on when bacteria reproduce. Okay. This is more time consuming. All right. So this is a slower process. Okay. So I hope that you have understood and I hope that you have enjoyed 1.3 viruses and bacteria. I hope that this can help you out with everything that you need. Have a great day and be kind and be happy and be healthy. See you later.